on, guys, hurry up. We have 5 minutes till broadcast.
Bear beats Battlestar Galactica.
No, Mom. I'm sorry, I've got to go. I have to do the news now. I'm very important in America.
Are you almost done with your makeup yet?
Son of a...
I don't know what you're doing.
Welcome to the Channel 46 Quibbler Nightly News with anchorman Victor Crumb, co-anchor and weather with Fleur Delacour, and chief investigative reporter Rita Skeeter, interviewing penguin researcher Dr. Welch. And joining the Channel 87 news team tonight is our science correspondent, Professor Horace Slughorn. And now, here's your #1 rated anchor for the past 2 days, Victor Crumb.
Good evening, citizens of Fargo. I am beloved anchor Victor Crumb.
And I'm your co-anchor and weather girl, Fleur Delacour, equally beloved as Victor Crumb.
Here's what's happening in your world today.
We have a special Channel 23 news broadcast coming all the way from Antarctica involving myoglobin in emperor penguins.
Breaking news. It's horrifying, devastating, heartbreaking, tear-inducing, stomach-sinking, ulcer-inducing. Breaking news update about Honey the Bee that was tragically addicted to cocaine.
Unfortunately, Honey checked out of rehab only to relapse back to her old cocaine habits. Honey the Bee is now dead. A moment of silence for her, please.
And on to other news. Channel 63's main news story of the night. New research has shown there is an increase in myoglobin in emperor penguins due to diving experience, age, and shivering. Our recent study of emperor penguins has shown myoglobin increases as penguins age. This myoglobin increase allows them to stay underwater for longer periods of time. A local citizen of Fargo voices his interest.
Emperor penguins hold their breath for a really long time. How do they do that?
That is the very question that we tried to answer here at Channel 33 News. The mystery of myoglobin is a topic that puzzled us here at Channel 34 News until a research team from Antarctica contacted us with breaking news scientific research. These researchers have found very interesting data about the ties between myoglobin and emperor penguins, which caught the eye of Channel 3 News. The researchers have an incredible interest in penguins.
I like penguins.
We will now have our science correspondent, Professor Horace Slughorn, explain myoglobin.
Thank you, Victor and Fleur. Hello. My name is Professor Horace Slughorn. I will draw you a rough illustration of myoglobin. Myoglobin. Myoglobin is one of the pigments in the blood that is primarily used for storage and transport of oxygen to the muscles. Myoglobin is monomeric, which means it has one binding site for oxygen. An increase in myoglobin is more important to increase diving time than an increase in hemoglobin because myoglobin is specific to oxygen. Hemoglobin has multiple binding sites that can bind to other molecules such as carbon dioxide. Myoglobin is mostly found in skeletal muscle. Back to you in the studio.
Thank you, Professor Slughorn. To understand this research fully, we sent our chief investigative reporter, Rita, to Antarctica to interview the head researcher that oversaw the study on myoglobin in emperor penguins. But first, what's the weather like in Antarctica?
It's a chilly day in Antarctica with a little bit of breeze. But I'm here today with Dr. Welch. Dr. Welch, tell me about your research.
Isn't it a gorgeous day?
If you say so.
We studied emperor penguins to learn about their myoglobin content and myoglobin mRNA concentrations, and how they change based on the age and diving experience of the penguin. We studied 10 wild chicks, some wild juveniles, 9 wild adults, and 5 captive adults. We took samples from wild adults and chicks, and then obtained some chicks from a colony to study them as they age to the point where they are able to start diving in a sea camp.
What do these chicks do at these sea camps?
Well, we supplied a 1 meter deep, 2 meter by 3 meter pool and a 16 meter long, 1 meter deep channel where the penguins were allowed to swim laps. We were able to train them with food rewards and have them swim up to 10 laps with less than 30 seconds submergence per day for 10 days.
What penguins did you sample?
The penguins we collected in August were obtained from the Cape Washington colony, estimated to be about 3 to 4 months old. Adult birds from the McMurdo Sound were collected and held at a sea ice camp for 2 months, after which we obtained muscle biopsy samples. The juveniles we studied were captured from the Cape Washington colony, where they were allowed to access to the diving pool until they learned to swim. After about a month, samples were taken and the penguins were released. The captive adult samples were obtained from penguins at SeaWorld in San Diego, 4 of which were transported as chicks and raised in captivity for 1 year and 1 3-year-old sub-adult which was born in captivity.
How did you determine their myoglobin content?
We took pectoral needle muscle biopsies and ran spectrophotometric and RNase protection assays.
What were the results of your research, Dr. Welch?
Well, we found that wild emperor penguins have higher levels of myoglobin and myoglobin mRNAs compared to the captive emperor penguins. These 2 factors also increased as the penguins grew older. The most interesting part of this research was the results obtained from the juvenile penguins. Before they learned how to swim, they still had significant levels of myoglobin content in their muscles. After they learned to swim, their myoglobin continued to increase. This is an indicator that dive experience and hypoxic environments increase the production of myoglobin. We also believe that transcriptional processes are the primary regulators of myoglobin production, but we need to research more, as there have been very few studies in this area.
Fascinating. Back to you in the studio, Victor.
Being the scholar that I am, I find extreme interest in the study of myoglobin in penguins. Not only do I have a love for the sciences, I also secretly love penguins to death. In order to fully grasp this concept, I must hear this from the horse's mouth. I have taken the liberty to talk to Minerva McGonagall, a scientist that is world-renowned in her studies, which pertain to myoglobin. I decided I would have a sit-down with this so-called expert and see what she thinks about this whole myoglobin dilemma. I feel my knowledge in penguins surpasses anyone else on this earth. So let's see if old Minerva McGonagall can teach a dog new tricks. Minerva McGonagall,
Hi.
pleased to meet you.
Nice to meet you.
Can I call you Mary?
No?
Let's have a seat. So Mary, why were penguins used for this experiment instead of other animals or even human beings?
Well, as my good friend August Crow, founder of the Crow Principle, would say, for every study, there's a perfect animal to find the answer. Penguins have one of the highest concentrations of myoglobin of any vertebrate. Also, increased oxygen storage is essential for them to be able to dive and progressively increases throughout development.
I disagree. I think it would have been a much better test subject.
I don't think you could hold your breath as long as a penguin.
I'm not sure how I feel about you as a person, Mary.
It's Minerva. Minerva.
How do penguins increase their myoglobin content, especially when they are young and don't do any diving?
Well, shivering and wing flapping increase myoglobin production in the muscles by activating the Calcineurin Pathway.
The Calcineurin Pathway? What is this evil magic you speak of, Mary?
I will first explain the general pathway used for most muscles to help transmit signals from the brain to the muscles.
Alright, let's hear it.
The pathway is first activated by an increase in calcium inside of the cell due to stimulus opening voltage-gated calcium channels. The calcium entering the cell causes the cell to depolarize, which starts a reaction in the presynaptic cell. Calcium signals to the vesicles to move to the membrane and release their neurotransmitter. This neurotransmitter, which is acetylcholine in the case of a muscle cell, binds to receptors on the postsynaptic cell, starting a depolarization to the muscle cell, resulting in movement. Specifically for the calcineurin pathway, the elevated intracellular calcium concentrations increase the expression of calcineurin. Calcineurin is able to dephosphorylate the transcription factor NFAT, which translocates into the nucleus and coordinates myoglobin gene expression, along with other transcription factors. Hypoxia and exercise, such as those encountered during diving, activate this pathway and therefore myoglobin production.
Sure, I see what you did. A complete regurgitation of Wikipedia. See, Mary, you and I, we think alike.
This information actually comes from scientific articles, documents that are published, and ideas challenged by the scientific community. I don't think information from Wikipedia would suffice for analyzing this report.
Well, I prefer Wikipedia for all my scientific facts.
I can see you being one of those people.
Let's move on, shall we?
This being said, we can see that the movement of shivering and wing flapping are most likely the stimulus that ends up elevating the SR calcium concentrations, leading to the calcineurin pathway and elevating myoglobin production.
I'm starting to make sense of this. So this is observed across all emperor penguins, am I right?
Well, this is kind of a yes or no answer. For instance, penguins in captivity, such as zoo-dwelling penguins.
And why would that matter?
Because these zoo penguins don't dive like their fellow penguins in the wild, the drastic increase of myoglobin will not be observed. There's a 60% difference of adult myoglobin concentrations when comparing wild penguins and penguins in captivity.
So one could say that these penguins are different, correct?
In some ways, yes.
So one could say that these zoo penguins are not real penguins, Mary.
Absolutely not.
When I dot the T's and cross the I's, this is my conclusion.
Everything you have just concluded is not accurate and in no way should be repeated.
Breaking news just in, Mary. Captive penguins, such as zoo penguins, are fake penguins.
I think what you mean to say is that captive penguins have lower levels of myoglobin, hence they seem fake. This is just due to the fact that they are less exposed to environments where they can dive frequently and for long periods of time.
Like I said, fake penguins. Are there any other animals that can be imposters?
Like I said, no animal is fake, but some seasonal myoglobin changes occur in muskrats and seals. Like penguin myoglobin concentrations in pigeons increase as pigeons get older.
And the truth shall set you free. Fake animals are overrunning the world. Thank you so much for your time, Mary.
It's Minerva.
Whatever helps you sleep at night. Back to the station.
So there you have it, everybody. Myoglobin and penguins. Tune in next week for our special of polygamy and mammals. For all of us at Channel 21 News, I'm Flor Delacour.